The title of today's webinar is Advancements in Nanopore Sequencing for Enhanced Food Safety and Quality Assurance. Our speakers today are Cameron Parsons, R&D scientist at Merieux Nutrisciences, Mark Allett, research microbiologist at the FDA Center for Food Safety and Applied Nutrition, and Selina Toh, Global Segment Marketing Manager for Food at Oxford Nanopore Technologies. So with that, let me turn it over to Cameron. Please go ahead.
So at Merieux Nutrisciences, we are actively involved in developing better food testing methods. And these are just a few of the applications that we've looked at where we have seen some distinct advantages for the application of long-read sequencing. So just a general overview, we're going to get a little bit better understanding of what long-read sequencing is, and then again, get into those applications that I had mentioned where we have seen some real value or advantages for long-read sequencing. Again, there's a lot of different terminology floating around the sequencing world. So just to kind of orient ourselves a bit, second generation sequencing is also commonly referred to as sequencing by synthesis. It typically involves, with the sequencing, an addition of nucleotide at a time, incorporating fluorescent dyes so that as those nucleotides are incorporated, there's a fluorescent signal that then gets captured. Typically, all these types of sequencing are dependent on amplification, and they'll generate read sizes, well, typically no larger than 400 base pairs, but in many cases, much smaller and more so in the 150 to 250 base pair range. So while the sequences are not all that large in size, there are millions or potentially billions of them. So, they try to make up for in numbers what may not be accomplished by being all that large in and of themselves. This is currently probably the most widely used NGS application in terms of sequencing. Then third generation sequencing is also commonly known as long-read sequencing. This potentially covers several different types of sequencing chemistry, which due to time, I won't get too in-depth on here today. However, the main takeaway here is that these more recent additions to the sequencing field typically have read lengths that are much, much longer. You see here, depending on your library preparation method, they can range anywhere from 4,000 to 300,000 base pairs. They're probably averaging more like the 4,000 to say 10,000, but that's still considerably larger than your short read technologies. And we'll get into a bit more specifics as to the situations in which it may be more advantageous to have these longer reads. So one of the first applications is shotgun metagenomics. For anyone that may not be familiar, this involves sequencing the total DNA content of a sample, whether that's a soil sample, an environmental sponge sample, an actual food sample. You're really not limited so long as you can recover quantifiable DNA from it. You can get a microbial profile. As you can see in that second bullet, it detects multiple types of organisms, whether it's bacteria, viruses, fungi, protozoa, or even higher order eukaryotes. So that's for food testing, you're typically more interested in the microbial community. So you can also, since it will detect whatever is present in a sample, you could use this to assess either the safety of a product by evaluating for pathogens that may be present in a sample, in this case, whether they're either bacterial pathogens, fungal pathogens, or viral pathogens. You can also evaluate the quality of a product by detecting spoilage microorganisms or things that may potentially cause problems down the line if they're either in a finished product or in an ingredient to be incorporated into a finished product. Additionally, with an appropriate amount of sequencing, it's possible to obtain strain level differentiation should multiple strains or genotypes be present. So this is just one example of some of the shotgun metagenomic data we've collected using long-read sequencing. This particular sample came from a blueberry. As you can see, there's a pretty diverse microbial community here, encompassing archaea, bacteria, as well as fungi and viruses. As I alluded to earlier, you can use this for monitoring your supply chain for agents of concern, either for quality or safety. And previous studies have, while you can do metagenomic sequencing with either short reads or long reads, there have been several distinct advantages established for using long reads for this application, such as getting more contiguous metagenome assemblies, which leads to more accurate taxonomic assignments. So the analogy I like to use is that if you're going to do a puzzle and you're trying to put together a picture, do you want to try to put together 1,000 small pieces or 10 larger pieces? So, since you've got complex microbial communities with potentially hundreds of taxa, if not more, then having larger pieces to work with can be a real assist, both computationally as well as for accuracy. Another application that we have evaluated long-read sequencing for is quasi-metagenomics. It's got a bit of a mouthful title there. It refers to the application of metagenomic methods like shotgun sequencing, but your matrix has undergone some sort of enrichment, either using a selective broth or even more advanced methods for trying to target and physically separate out your organism of interest before you do the sequencing. As you can see there have been quite a few studies looking into this sort of application. While there have been, as I said, had been multiple studies, so there are multiple approaches to doing this. Outlined here is the approach that we used for our evaluations. So you started with a sample of interest. I'll get more into that on the next slide. They underwent a microbial growth enrichment in RV broth. Our target organism for these studies was salmonella. So we had immunomagnetic separation beads that specifically targeted salmonella. So any salmonella present in the sample should theoretically bind to the beads, which could then be pulled out magnetically. And then multiple displacement amplification means that we were then taking out those magnetic beads, using a lysis treatment on the cells from the beads, releasing their DNA, and then amplifying whatever DNA was present in the sample, and then sequencing it on the Oxford Nanopore mid-ion. So for our studies, we used two different matrices, lettuce and dry dog food. We inoculated each sample with roughly 1 CFU per gram of salmonella using two different serotypes, or one distinct serotype per product, but two different serotypes, and then subjected them to the quasi-metagenomic method I just described. So they were enriched, and then salmonella were separated and sequenced. In both cases, genome assemblies from the quasi-metagenomic sequencing matched identically to the inoculation strain, meaning that these products that had a relatively low level of salmonella on them the salmonella was detected, not just detected, but also the sequencing products were identical to the salmonella that was placed on them, showing that without any sort of cultural isolation, you're able to get good quality sequencing rapidly from a direct enrichment of the product rather than having to go through all the time-consuming isolation and purification steps, which I'll get more into for our next application. So again, this is targeted. So in many cases, pathogens are present in very low abundance in food products. So that can be an impediment to being detected with a variety of methods. So this allows you to go in, physically separate out those organisms and get good quality sequencing. Based on that, since you don't need to go through those isolation steps, as I already mentioned, this can be much faster. And in the cases with natural products, there are many times where you can have multiple strains or multiple genotypes of your organism of interest. So if you go through the isolation steps and then after all of that, just pick a single colony to analyze, it can tell you that, oh, you have this one type of this organism, whereas you might actually have multiple types, whereas this approach would then also tell you, hey, you don't just have one type, you have multiples. Or in the case of like an outbreak investigation where you're looking for a specific genotype, this can be particularly important, as if you just look for one, you might miss the one that you're actually interested in. Which kind of is a good segue into the next application, focusing a bit more on the safety side of things. With foodborne pathogens, it's really key to then be able to subtype them to know, are these strains, or just because you have salmonella, is it the salmonella you're looking for, again, in the case of an outbreak, or a recurring strain? So you need to have a way to differentiate them to be able to tell that. So again, most typical schemes, you start with a test portion. It's going to involve several rounds of enrichment and then further plating on selective media to get down to an isolate from a complex food sample. Each one of these stages indicated here can take roughly 24 hours, in some cases more. So there's that time element, and as can be the case with selected media, different subtypes of different pathogens can have different growth potentials. So they may outcompete your subtype of interest, and you might not actually recover your problem strain or your problem subtype. So just because you isolate it out doesn't mean you've again gotten your problem organism back out. So the go-to method, again focusing on salmonella, is serotyping. So this involves anti-seroagglutination to distinguish different serovars of salmonella. So in addition to the isolation process, which I just described, traditional serology can extend that identification period by an additional 2 to 3 days. So in some cases too, if you're then going to do whole genome sequencing, that can require 10 days or longer for isolation, library preparation, and sequencing, depending on various other factors. With the application of third generation sequencing, that period can be decreased to 24 to 48 hours from receipt of an isolate. So you can then potentially use sequencing to do the serotyping approach for salmonella. So our workflow for testing out, trying to do this serotyping approach is to go from an isolate, DNA extraction, library preparation, on through to sequencing. I know I mentioned the speed of it, though, from this graphic here. It may look a little unwieldy, but quite a few of those steps happen pretty quickly. So for our evaluation, we were comparing the results of classical serology of salmonella with the aforementioned molecular serotyping method involving Seq02. We see that in silico serotype predictions were correctly made 90% of the time, whereas classical serology did have an element of misidentification, since that involves a human evaluating anti-sera agglutination, that can be a little bit of a subjective process. We do see that there were still some misidentifications with molecular serotyping, as there are some closely related serotypes that are just kind of difficult to differentiate even with good quality sequencing data. And there was at least one that was undetermined. So any method is probably not going to be 100% foolproof, but in terms of molecular serotyping, it's rapid and it's accurate. And one of the things we had also found with our evaluations is in many cases, you could get correct serotype predictions after as little as 30 minutes of sequencing. So, some of those second generation sequencing applications, you have to wait for the completion of a sequencing run, which can take 20 to 36 hours. Whereas with the third generation, again, you can run it for as little as 30 minutes and get the information you need. So for salmonella, molecular serotyping can be more rapid and more definitive, less susceptible to user error or interpretation. And these analyses can be run from either whole genome metagenomic or quasi-metagenomic sequencing data. So it makes them pretty versatile in terms of the analytical workflows they can be applied to. And they can also alert you if you have those multiple genotypes in a sample to let you know if additional analyses are required or additional sampling, whereas traditional serology may not give you that information. So metagenomics with long read sequencing can be more in-depth and accurate. Quasi-metagenomics can be faster, more accurate, and again, really let you drill down into the true population of your organism of interest, as opposed to if you're biasing that by just taking single colonies. And sequencing can provide deeper insights and let you test for a broader array of things with a single test. So while in some cases it can be more expensive if it's replacing multiple tests, both in terms of time and the depth of information you get, it's definitely well worth it in terms of food safety and food quality. So with that, I will turn things over to Dr. Allard with the FDA.
Hello, and thank you. We'll get started here. I'm going to be talking about the FDA's perspective in use and encouragement of using genomics for food safety. FDA has been actively assessing and working with Oxford Nanopore technologies since 2017. And essentially, as the research and development arm of the FDA, we buy and test equipment. So we've tried a lot of these different equipment, and over the years, multiple chemistries, software, as well as sequencing instruments. And essentially, and Cameron's already covered a lot of this, you have with longer read sequencing, you can get more accurate assembly. We find these high quality reference genomes very important for SNP analysis, because if you have more of the genome resolved, even a complete genome, then you usually can call more SNPs accurately, which can add evidence to your phylogenetic work. And this can help you understand long-term evolution, comprehensive view of the genetic composition. In particular, we're very interested in mobile elements, plasmids and phages, and genomic islands. We also can predict gene presence like virulence genes or antimicrobial resistance. And then the additional evidence is this technology can also look at DNA modifications if you're interested in that. And so what we're talking about is going into the farm to fork continuum of food production, and you might sample and find a positive foodborne pathogen in any of the parts, starting from ingredients to some processing plants, manufacturing. And essentially what you want to know is what we call trace forward and trace backward, which is you've found a pathogen, you've characterized it, where did it come from, and essentially where else did it get shipped to? And this to fully understand the risk and to develop preventative controls, you really need to know where the pathogen and the contamination originated, and you need to know if it's the same thing or something new. And genomics can support this. And so FDA, we've been closing bacterial genomes and making these publicly available for some time. We have over 1,000 complete bacterial genomes that were assisted with long-read assembly. And the majority of these are salmonella, but we've looked at a diversity of foodborne and human pathogens to primarily bacterial. These are all publicly available. And just to show you, here's a fly assembly graphics, the importance of completing the bacterial genome, and in particular, any mobile elements, plasmids, or phages. And so the short read sequencing typically has a lot of trouble, particularly with repetitive elements. And the longer reads, the longer the reads, the more likely you can fully assemble the pathogen that you've collected. So I want to talk about determining pathogenicity in E. coli, and the National Advisory Committee on Microbiology Criteria for Foods, the NACMIF, is a FDA, USDA, CDC advisory group, and they meet regularly on important foodborne pathogen issues that they need to discuss. And they came out some time ago with STEC virulence. And what you see is, depending on the genes present, there's an increasing risk that these will be, harmful pathogenic bacteria that cause human disease. And so we are using to assess the risk of an isolate that you've collected, you want to look to see if these genes are present. And traditionally, they've been worked on by PCR. And these related to the Shiga toxigenic genes, the STX1 and STX2, but also whether there's attachment genes involved. Because if they can attach, then the toxin is in place and there are more bad outcomes and higher pathogenicity. So with long-read sequencing, of course, you get all the genes. So you don't have to do multiple PCRs. You can do a single sequencing run. You can get all the presence, absence of genes. And then you can also replace other kinds of tests. So there's other ways to subtype. Historically, this is MLST, serotype, virulome, then the genes present and absent, like STX1 and 2, and the EAA and AMR typing. Well, all of this can be pulled out of the genomic data from long-read sequencing, the gene presence. And of course, you can do comparative genetics, which is building a phylogenetic tree and seeing if the characterized pathogen is linking to any clinical or previous isolates in the public domain. But essentially, this idea that the genomics can replace multiple subtyping tools will save money and time. And the accuracy for most of these systems is very high, in the high 90 percentile. Though I would say there's still time to do both. And to do like AMR prediction, it's good to have both the genome and the antibiogram, to essentially be able to predict better genes, phenotypes from the genotype. And the other thing is, I think we're moving the whole food safety and microbiology in general is moving from genes to operons to metabolic pathways and then to systems approaches. If we can really understand how the bacteria are surviving in their environment, how they're competing, then you can use that information from genomics and in fact from microbiome as well. You can use that information to understand how to control. And this is also happening in the hospital, in clinical diseases, is understanding the infection and the toxicity, essentially what causes the bacteria to go into these cycles. You could prevent them. And the same is true. We think we could use this for preventative controls in a chicken house or in a cheese manufacturing facility where you might be able to control, more knowledge provides you with more information to design preventative controls at work. In particular, what we're most concerned with is resident versus transient pathogens. Transient pathogens aren't really a problem. They pass through the facility. They may contaminate some food, but they're here and then they're gone. They don't usually affect large quantities of food. It's just the regional piece that got contaminated. But when you have a resident pathogen, either in the field, and this could be, say, a large animal operation nearby, adjacent land use, it may be the fertilizers and soil amendments that are coming from those facilities. If it's a common and a persistent pathogen that's resident, when that food moves into the manufacturing and processing facility, it can establish, some of these can establish inside the facility. And then once they're there and they're slowly leaking out, essentially this produces a lot of contaminated food, which makes a lot of people sick. And so what you want to know when you genomically characterize the pathogens is to understand whether you've seen this before, whether they're returning, or whether they've never been, you may have knocked down in your preventative control, you've knocked it down, but you haven't eliminated it from the facility. And so genomics can help you identify whether you have a resident pathogen and whether your preventative controls are actually working. Some of the time, it's not apparent what's going on and how the contamination occurred, that you weren't able to solve the root cause of the contamination event. And in this case, we have to do what are called longitudinal studies. And typically, we don't get this as multi-year assessment of the whole environment and to get a full picture of where the pathogens are surviving in the environment and how they're moving through the environment and contaminating food. And so we've done this in this particular example. What this was understanding a reoccurring outbreaks in tomatoes on the Delmarva Peninsula on the Eastern Shore. And what they found is that through multiple sampling, the red shows where they regularly find salmonella, and it's in the standing fresh water. And so if there's standing fresh water, at literally any time of year, you can go and sample and find the salmonella pathogens that have been highly adapted to these environments. And so by advising, essentially using that information to inform the farmers about good farming practices, in particular in their use of this water source, essentially saying don't use fresh water from standing fresh water because it's contaminated, use well water or sterilize the water in some way prior to critical points of when the plants are first planted into the field, and also through use of things like pesticides and herbicides. So by providing the guidance of the root cause, industry can make their product safer. And here's just a particular example. This is a peach orchard. And of course, FDA advises all produce manufacturers and growers that you have to be aware of the adjacent land use of your facility. And in this case, this particular peach orchard has a poultry broiler house and production on one side of it and a dairy farm on the other side of it. And they had outbreaks. And essentially what was happening is that the outbreaks, the pathogens were moving from these animal facilities to the peach orchards. But where they found the pathogens wasn't in the middle of the orchard. It was just right at the very edge, essentially the hedgerow. And these careful studies of these sequencing and analysis of these pathogens essentially show the significance. One is that it documented carriage of the pathogens in fugitive dust samples. So the animals, their waste is on the ground in a drier environment. They get into dust, and the winds, prevailing winds, can blow those dust into the orchards, and then they would contaminate the fruit. So this essentially provides, just like the freshwater advice, this provides understanding of roles of buffers and windbreaks. It also documented, we did this inside the phytotron, where you actually did internalization studies to see if you could blow contaminated dust onto peaches and see them essentially internalize during flowering the pathogen. And so this helps us understand a lot about peach safety, which is written into good farming practices to help industry. And it was genomics that essentially uncovered the details of how the pathogen's moving through the environment. Essentially, the genomic links can incentivize food manufacturers to invest in preventative controls. If you don't know you have a problem, you're not going to fix it. And so this is additional genomic evidence to show directly how the pathogens are moving. And once you know the root cause of the investigation, this improves knowledge of reservoirs, risks, and working preventative controls. And then the food industry can track this. They want to produce safe food, and the FDA Office of Compliance also watches these databases very carefully, looking for harborage and reoccurrence to see if the contamination is under control. And so genomics is an important tool that industry should adopt and use to understand reoccurring problems. I just want to say it's not just the genomic data, it's the metadata. The description of the data can help in analysis. And we talk about fair practices, findable, accessible, interoperable, and reusable. The idea is you collect a genome once, upload it to the public domain, and this way many other people can reuse that data to understand their connection to the pathogen. And so FDA does this in a transparent way. We have all of our protocols are listed in protocols.io. They're publicly available so that you can understand exactly how the government does every step from gathering the sample, extracting DNA, the sequencing and analysis. And the analysis in particular, we use a cloud-based program called Galaxy Tracker, which is an instance of Galaxy. And this way we can share working and validated data analysis pipelines to analyze the data rapidly. This way people don't have to construct their own pipelines. They can use, if they wish, they can use the known validated analyses. And then lastly, all of our data is put into open repositories at NCBI and INSDC. And these are embedded. They're actually present both at FDA and at NCBI in their biosample. And the idea is we should standardize language of what we're collecting so we're all talking about the same sample. And this includes the on-the-farm environments. It includes food production environments. It includes the animals themselves. And so this helps to standardize use. And so in particular, we have, it's sort of, here's a schematic of a model illustrating the logical relationships among unique terms representing different information facets used in describing a swab inside of a facility, like a conveyor belt. And I have another slide. Essentially, what we found is when you don't standardize, if you let 10 people describe the red square on the left, they'll all describe it a little differently. But if you give them a pull-down menus where you're saying, what's the structure? What's the sub part of the structure? What's the position that the swab was taken on? What's the material, the actual material it's made of? And then what's the condition of the material? Like, is it rusted or encrusted with food? And essentially, if you provide standard pull-down menus to inspectors, they'll all describe it exactly the same way when you give them a structure. And then on the right-hand side, what you can do is do rapid analysis of, hundreds or thousands of samples, and essentially say you can do things like prevalence of the pathogen. And I want to just point out on the left-hand side, these are the most commonly sampled and swabbed places from an FDA inspection. And you see the green is when they find positive. So we know where to find stuff. But on the far right, here's an example where a few investigators sampled brooms, but they're highly positive. And so this would be an important sentinel for a facility. How long have you had your broom? How often is it used, and which rooms does it go to? And is it ever cleaned, or is it ever tested? And what we find is that, you cover a lot of ground with your broom, and so you're more likely to pick up contaminants inside of a facility. But you want to be a cleaning agent, not a swabbing and distribution tool. So we can learn a lot and this will feed into machine learning and artificial intelligence. The genomics has a lot of prediction power, and you just need to tie good metagenomics to really make it more useful. So all of our data from essentially anyone who's willing to upload their data and make it public. At the FDA, we push data to PulseNet, which is the CDC primarily clinical database, but we add the food and environmental data to that database, as does the USDA. And we also send it to NCBI pathogen detection, and that's globally shared every night through the INSTC. So that's all of the known publicly available data. And then the different members of the INSTC have tools. So for example, NCBI pathogen detection provides daily clustering of pathogens for under 50 SNPs or less, and they do rapid calling of all the genes. Any genome that's uploaded automatically has antimicrobial resistance, virulence, pathogenicity, and stress genes. And then, of course, the data is in a structured way, so you can use your own dashboards to essentially pull out data and do analysis of it. Essentially, genomics and long-read sequencing provides greater certainty when matching clinical, environmental, and product isolates. It provides the link between isolates and potential sources of contamination, and essentially it allows investigators to do things in a more targeted way. And the faster products recalled, the fewer people get sick. And I just have one more slide. There's economic benefits to industry. Here's Gunther et al., which is a New Zealand academic group. They've documented case studies of millions of dollars per year, over $100 million annually to food producers in New Zealand. A lot of that's cattle and kiwis. And it's basically preventing outbreaks preventing recalls, and allowing markets to expand. And the FDA did similar economic modeling. And what they showed, this is from a reduction in averted illnesses by using genomics to rapidly identify outbreaks, that we estimate today that we're over $1 billion of annual reduction in human illness. And this has been increasing. Essentially, it says for every dollar invested, you get $10 of averted human health cost. And the industry would be getting probably a similar, based on the New Zealand work, a similar value in adopting genomics to help prevent problems down the line. Thank you so much. Our next speaker is Selena Toe.
Thank you, Mark. Appreciate everyone for joining us today. To follow up on what Mark and Cameron have already touched on, there's a lot of great use cases and applications that we support to deliver the future of food genomics with nanopore sequencing. Food safety and quality encompass a range of nanopore sequencing techniques, and the approach you use depends on your research or QC test objectives, and it can range from foodborne pathogens or spoilage detection and prediction that you heard of early on in the webinar, and also food authenticity and even probiotic and starter culture strain engineering. Now, unlike legacy NGS methods, we listen to the voice of the food industry and offer capital-free, scalable instruments with an option to lock the technology with simplified workflows, some of which are end-to-end, with flexible sample batching and real-time analysis with our Epitome software, all of which allow labs of all sizes to be able to bring nanopore sequencing in-house. And that is what motivates the community, which involves food companies and public health and regulatory agencies to evaluate or implement nanopore sequencing into their research or QC operations, since their features best align with the goals of timely microbial risk assessment and root cause analyses in food safety and quality to generate actionable insights. And to accommodate the various skill set within the food industry, most don't come from molecular biology background or genomics background. And this helps reduce human error and shorten learning curve, in which motivate us to develop end-to-end or semi-end-to-end workflows, shown on this chart, which can all be adaptable to collect additional test outcomes that may be of interest to you. As such, users are now able to reproduce the accuracy and data that we've benchmarked using samples that are credible and relevant to the food industry. So samples from FDA CIFSAN, ATCC, Martin Weidman's lab from Cornell, and or the Zymo mock community. One of the end-to-end workflows that I want to highlight is NOMIS, our nanopore-only microbial isolate sequencing. This is the fastest wet lab with the push-button epitome analysis to automatically generate genus species identification, salmonella serotyping, gene annotation, subtyping, and antimicrobial resistance. And the most exciting part of this launch is our integration of Seq02 into our epitomes analysis, a pipeline developed by University of Georgia and CDC PulseNet that is pending addition to the FDA BAM to be the new gold standard for salmonella serotyping. As most of you know, the FDA BAM is well respected and adopted by researchers, food companies, and public health labs across the globe. And we do intend to look at how we'll continue to innovate and streamline processes by aligning with the FDA BAM and the smarter era of food safety, USDA MLG, and others where we can make the most impact in protecting our food supply chain and public health. And with that, I'll hand it back to Julia to facilitate the live Q&A session. Thanks again for listening, and I look forward to our roundtable discussion.